in this video, I'm going to talk about the structure and function of the eye. So some basic anatomy. The cornea is the most anterior component. It's a clear covering that we see right here in the picture. It's the clear covering that is anterior to or superficial to the pupil, iris, and the lens. So it's protecting those structures underneath. So it's clear like a window. So it's allowing light to shine through and enter into the pupil. And so it's not opaque. Whereas the sclera, that is outside the cornea. So that's, it's lateral to, superior to, inferior to. So it's the whole white part of your eye outside of where the cornea is covering the iris. Okay, so the cornea is that clear window-like part that's covering the pupil, iris, and the lens. The pupil is the actual hole. It's the black dot right in the center. It's the hole that allows light to enter into the eyeball. So the diameter of the pupil increases and decreases depending on how much light is in the surrounding environment so that we can adjust how much light is permitted to enter into the eyeball to make a clear focused image. The iris is the color part of the eye that surrounds the pupil. The lens is back here in our picture. That's the lens. It's behind the iris, and it's a transparent structure that allows the eye to focus at different distances. So it's sort of like if you wear glasses or contacts, those are lenses where the idea is that they are refracting the light in different ways to change the focus of the image. So that's what the lens is doing inside of the eyeball. It's changing in shape, essentially. It kind of gets squished and it gets stretched out and it changes in shape to help to focus on different things that we're looking at based on whether they are up close or further away, so that it can create a focused image on the retina and the posterior wall of the eyeball. And so it's when the lens is not doing that effectively or not well enough that then we add corrective lenses. So we'd add contacts or glasses to help make up for the difference in what the lens, the eyeball is not doing. And then, as I mentioned, the sclera is the white of the eye. So it's surrounding all of the other structures and it is opaque. It's white because it includes a lot of collagen, and collagen is white. It helps maintain the shape of the eye and protects the inner structures of the eye. And then the conjunctiva is a mucous membrane that protects the covering of the sclera. So like if someone has conjunctivitis, that would be inflammation of the conjunctiva, that's pink eye. So it's a bacterial infection that's causing inflammation of the conjunctiva. So then looking inside of the eyeball, we have three chambers or three kind of sections within the eyeball. So the anterior chamber is all the way anterior. It's between the cornea and the lens. It's filled with what's called aqueous humor, which I'll describe in a second. Then the posterior chamber is right here. And that's between the iris and the lens. It's also filled with aqueous humor. So the anterior and posterior chambers are connected to each other, and they're both filled with the same fluid called aqueous humor. It's clear fluid, and it has to be clear because we need to allow light to pass through and to transmit the image to the retina. But it's clear fluid that nourishes the cornea and lens. It's constantly replenished and replaced about every 90 to 100 minutes. Okay, so it's constantly cycled out so that we are sending more nutrients and oxygen to those structures and carrying away waste as we're carrying away kind of the used up aqueous humor as it's replaced. The vitreous chamber is the posterior, it's filling up the posterior portion of the eyeball. So that is the large space posterior to the lens. And it's filled with what's called vitreous humor. It's also called vitreous body, and it's really just like a big gelatinous ball, and that's what fills up the main space of the eyeball. It is not actively regenerated. It's pretty much immobile, and it just, you've got this big kind of portion of jelly in the eyeball, and it just stays there forever, basically. It's clear, again, because we need to allow for light to be transmitted through it. And it's not going anywhere. Okay, the retina is this yellow layer here in this picture. Lines the back wall of the eye, and it contains lots of different neurons and photoreceptors, so sensory receptors. The fovea centralis, sometimes just referred to as the fovea, is the spot on the retina where objects seen in central vision are focused. So here is the fovea on the retina. Okay, so the light shines through, it comes through the hole, that's the pupil. It's modulated by the lens, so it's refracted by the lens, and then that image is sent and shines right back here and comes to a focused point right on the fovea. And that's where central vision takes place. The optic disc is this whole little area where the axons of the retina are kind of converging and coming together to form the optic nerve. So we're collecting up all of this visual sensory data, and that's all coming through and converging at the optic nerve or at the optic disc, which then becomes the optic nerve and is transmitted to different parts of the brain. Then we have rods and cones. Those are specific types of photoreceptors, and there's different types of each. But there are rods and cones. Those are photoreceptors that are part of the retina, and those are sensory receptors that are detecting light. So rods are detecting low levels of light. So they're responsible for night vision, so like grayscale vision. And they're located in the periphery of the retina. So they're throughout the retina outside of the fovea. Okay, so they are giving us peripheral vision because our central vision is occurring at the fovea, and that's where we have a higher concentration of cones. So cones give us color vision. They're responsible for color vision, and they are located mostly in the fovea, mostly at the center of the retina. So they give us central vision and visual acuity and color vision. Okay, so refraction. So the light is shining in through the pupil and it's going through the lens. So the cornea and the lens are both serving to refract the light, which is like bending that stream of light. And it's being bent so that the light comes to a point right at the fovea. And the fovea is located in a larger area called the macula. Okay, so the macula is this larger area that's central in the retina, and then the fovea is located within the macula. So we want that light to come to a perfect point and land right on that fovea, and that's what gives us our central vision. So the light needs to be refracted so that it can achieve that. So depending on how far away the thing is that we're trying to look at, we need the lens to kind of squeeze together or stretch out further. You know, we need it to change in shape so that we can achieve that little point of light right on the fovea. Okay, the optic nerve is cranial nerve number 2. So it's transmitting the sensory information from the eye to the brain. It's formed by all of the neurons that make up the retina. Their axons converge and come together at the optic disc and form the optic nerve that then travels posteriorly in the brain and heads to the occipital lobe and a couple other, detours along the way, different parts of the brain. Some need direct visual input. The optic chiasm refers to where some of those neurons that are traveling in the optic nerve flip and cross over to the opposite side. So like if we look at the visual field, the purple here is showing the left visual field and the orange is showing the right visual field. So each eyeball, the right side of the eyeball is seeing the left visual field and the left side of the eyeball is seeing the right visual field. Okay, so both eyeballs are collecting data from the whole visual field. But then we separate the left and right visual field so that we have it essentially flip-flopped in the brain, but it's organized by side. Okay, so all of the information about the right visual field is crossing over so that it's traveling to the left side of the brain, and all of the visual information from the left side of the visual field is crossing over so it's being received by the right side of the brain. And so that crossing over whereas basically half of the axons from each eye are crossing so that the left and right visual field is all organized. That crossing over is the optic chiasm. All right, thank you so much for watching. I'll see you in the next video.